hopefully you can take a look at it. It's about 20 minutes long. So yeah,
Oh,
it's
okay. And uh, I think I heard that someone won a Nobel Prize.
yeah, yeah. Just last year, some scientists in collaboration with Google developed this program called AlphaFold. So that's what the video is about. Yeah.
Yes, and we touched on that a little earlier in our discussion, right? How it's incumbent on cells to very carefully control the mutation of genes that encode proteins, because if you go too far from what you already have, you might run into proteins that will never fold. And so the ones you find in cells, I suspect, are, someone put it this way, I don't recall who said it, are basically a polynomial time foldable subset. Okay. Now, Martino, do you know if this AI program that did the folding works really well on naturally occurring proteins, meaning ones that cells use, and you can't count on it if you just give it a random sequence of amino acids?
Yeah, well, I remember the examples from the mini documentary, I guess, that I just sent you again about it. Um,
Yeah.
the examples involved some, so there's apparently this competition that's existed for over a decade. It's called CAT,
Okay.
and it's basically a competition for um, scientists across the country and the globe to every year there's a an event where a primary sequence of amino acids is given to all the contestants. It's publicly published, I guess. And then everybody in the competition using their own computational methods tries to synthesize the tertiary structure of the protein, and then they
Yes.
compare it to the, I guess, crystallography confirmed structure. Um,
Yes.
and then a passing score is considered anything over 90% accuracy. And for the past 2 decades that this competition has existed, no teams all over the world have been able to surpass that 90% figure. And AlphaFold was the first, I suppose, contestant that did that.
Now, just by the way you described uh, the trial sequence that they came up with,
Mhm.
uh, I am virtually certain that it came from nature.
Yes.
Okay. And that if you'd given it a random sequence, nobody could do anything with it. And I think that's because the ones that come from nature are quite special. Okay. They fold quickly. One of the reasons I say this is X-ray crystallography. Okay. That works by, let's say you wanted to take a protein and use X-ray crystallography to get a, I don't know what the current standard is, 4 angstrom resolution view of that folded protein. Okay. Now, what would happen uh, is it would never fold. Okay. That is, if it was random, it would have almost no chance of ever folding. And in your lifetime, for example. And as a result of that, it wouldn't fold into little well-formed things. And it's only proteins that fold into nice little things that you can now crystallize the stuff out and use X-ray crystallography on.
Hmm.
So you wouldn't have an X-ray crystallographic um,
Standard for,
standard. Yes,
yeah.
nice. Okay, so I think what's happening is the reason these AI programs work, or these programs, is because they are only challenged with naturally occurring proteins. And if you give them a random sequence, just, make up a 200 long amino acid sequence and give it to them, they won't have a clue. Okay. And the reason for that is if they had a clue, I could solve NP-complete problems. So just another one of these stories. I used to have a molecular biology lab at USC. And it came about because of my work on DNA computing. All right. So one of the questions that a little bit philosophical was, what is a computer, right? So one of the examples I gave was I happen to have a couple of stories up from my lab, Michael Waterman and his group. And Waterman was doing computational biology, where you're given a genome and you're supposed to work out things. And in his group was also, as I recall, people who could do protein folding. Not very well, but that's what they were aiming at, right? Okay. And but across the hall from me was an X-ray crystallographer. Okay. So in theory, I could have taken a primary sequence of amino acids, meaning just, I tell you this, you can know what I mean. And I could have run upstairs and asked Waterman's group to tell me how it folds. But I could have also walked across the hall to the X-ray crystallographer and said, here's the primary sequence. Would you tell me how it folds? All right. Now, I could ask them to give me the folding to a certain degree of accuracy. And if I made that sloppy enough, maybe they could actually get something. Okay. But I could have asked them, when you get the answer, slide a memory stick under my door, and I'll stick it into a standard program that somehow takes the standard result and shows me the folded thing. And I could have asked the person on the third floor, or I could have asked the person across the hall that was the X-ray crystallography to do the same thing, right? And if the person on the third floor had come up with a folding, we presume he'd used a computer to do it, right? But the person across the hall wouldn't have used a computer. He'd have, used chemicals and solutions and, okay, refrigerators and stuff like that, and X-rays. But from the point of view of input output, I would just get perhaps identical memory sticks. Yes. And so we would think the one that went upstairs, computation had occurred on the primary sequence. But the one across the hall, isn't that computation as well? Yeah. So
A
this has
different
to do
form.
a different
Yeah.
form, yes.
Yeah.
But computation is ubiquitous in the universe. Okay. It doesn't have to take the form of your laptop, right? It doesn't have to be silicon based. It doesn't have to, be made of plastic and other stuff like that.
My
All right.
question to you is, on the other hand, what do you think is happening? Well, so to give some background, in this previous Nobel Prize that was awarded in chemistry, it was actually split in half. And half of it went to the group that developed this AlphaFold AI program, which resolves the tertiary structure based off primary sequence of known extant proteins in organisms. However, the
Yes.
second half went to this group that developed another technology and I forgot the name, but I just looked it up. It's called RF diffusion. And what they're
Okay.
able to do is they're able to design primary sequences to achieve a desired tertiary structure. So they feed the algorithm a three-dimensional structure that they want to form with their protein. And
Yes.
then the algorithm, I'm assuming with its knowledge of existent biologically present proteins, then designs a primary sequence that will, I would assume, spontaneously fold into that conformation. So what do you think is happening there?
Yeah. Well, I haven't contemplated, so whatever I'm about to say is just going to be, mind sand uh, about it. So, okay, the input now is a primary sequence where, no, no, not a primary sequence, is a folded protein but presumably a bunch of little balls that have that are on a string and they're in a certain shape, but one doesn't know which amino acid goes in
Or at
each
which
ball.
points in the sequence? Yeah.
Okay, so and now the output's supposed to be fill in the balls, right? It's
Yeah.
sort of like a very complex Sudoku game or something. Where you have a bunch you're supposed to fill in the numbers, the digits 0 through 9. So it sounds to me like it's Sudoku, base 24, and it's much more complicated.
If
Okay.
you if
Yep.
you wish, I can see if I'm able to share my screen and show you that even that section of the video. It takes maybe 3 minutes. But I don't know if you want to allocate class time towards that, but maybe it would give you a better understanding of what they're doing.
No, okay. Um, I'm inclined not to do it right now,
Okay.
but I certainly want to see it, and maybe we
Okay.
will do it right now. But my initial thought is this is a Sudoku game, if however you say that, and you have to fill in one of what are there 21 amino acids that occur in nature or something like that?
20. Yeah.
20. Okay, you have to fill in the little balls uh, with amino acids. Now, uh, can you do that? Well, yes and no. So, um, because naturally occurring uh, DNA, some of its proteins have been crystallized, and we know their actual structure. Even when I was more into this, world, they had discerned repeated motifs, beta sheets, alpha, whatever they were called. Okay. Certain things that recurred, which makes sense because the cell has to keep, some structure. And I can imagine playing the game where you look at the uh, proffered Sudoku problem and you see structures like beta sheets. So you might know how to fill those in. And so uh, it wouldn't surprise me if you could succeed at that. However, if I give you something that a structure, if the input is a structure that has never been seen, and nothing like it has been seen naturally, they're not going to be able to fill it in. So that's my opinion. But what did they say, uh, Martino?
Well, I from the video, as you will see, they they explain that this is a technology which has been enabled by the presence of AI, but for a surface level explanation of it, they don't go into real depth about how it's done. So I was wondering if maybe you could, like, just out of intuition, maybe explain what,
Okay, okay.
yeah.
So let me explain. Um, so I'm going to try to make an analogy. Okay, if an architect drew a building, okay, and uh, and he even gave you blueprints for the building, except that he just tells you things like, we'll use some material to build these structures, and that material has to have certain, physical properties like, be able to resist compression and stuff like that, right? So he doesn't tell you use steel here, right? He doesn't tell you uh, use windows here. He just says, I want something here and it should allow light through, but not wind. Okay. Blah, blah, blah. And as long as he describes his building in this fairly precise, but hasn't told you what materials are needed at each position, if you're a good, you know, uh, architect, you can fill in, use steel here, and in particular, this kind of steel with chromium and blah, 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 right? He could fill it in for you. Now, if you gave him some structure that looked not remotely like a building, okay, but still a structure. I mean, I can lay down three-dimensional prints, and it is constructible in the sense that a three-dimensional object could be made that is doesn't violate any rules of physics or combinatorics, and but it was really, really weird. I don't know. An architect probably wouldn't know how to fill in the blanks. So, that's my mumble. Okay. So I think once again, there's probably the common theme that you can succeed if you're going to use a small sort of polynomial time subset of inputs. You're going to restrict yourself to that, then you can probably succeed. Okay, lots of people have ideas. So uh, thank you, Martino. Uh, Parku, Porku.
Yeah, close enough.
Okay, thanks, Martino. Okay, catcher.
Hi, Dr. Alman. So I don't know if you saw, but there was actually recently another uh, model released on this problem of protein folding, I believe among other things, but it's called EVO 2. And it's from NVIDIA and this new research institute called ARC Institute. But
Okay.
basically, you kept talking about synthetic primary sequences. And so this model, where AlphaFold treats protein folding on like a per case basis. It has like, I don't know, an algorithm or whatever to determine a single protein. This is more like an LLM that it trained on like something like 9 trillion base pairs from genomes across like eukaryotic, prokaryotic organisms. And basically, it's able to generate synthetic DNA sequences that it can predict what the protein folding will be based on, its training data from all these structural motifs like you were talking about.
Yes.
Uh, so I don't know if that like kind of contradicts what you're saying, or it might even agree with it because an LLM is, going to notice those patterns that you were talking about in, protein folding can only occur through certain pathways. But
Yeah,
so
yes.
yeah.
So I get it, catcher. And once again, they went to these databases of sequences, which already tells you that they're restricting their attention to naturally occurring objects, right? And naturally occurring objects have, because they're natural, meaning they occur in nature, typically, well, anyway, in the case, they're the ones that occur in nature are often a very constrained subset. So I'm guided by something that, you theoretical computer science should understand pretty clearly, that if I wanted to factor a big number, I could go in my room because, I'd have to figure out the reduction and all like that, and give you a primary sequence of amino acids. And from its minimum low energy folding, or whatever, I could find out how to factor the number. Okay. I mean, that's what our discussion earlier on this topic said. It's an NP complete problem, the protein folding. You have to, as a theoretician, encode it in a certain way to see that. All right. But if I could actually get the folding of an arbitrary sequence of DNA, I could solve 3 SAT problems. So if you can get that with a computer, you've solved and do it in polynomial time, you've proven NP is equal to P. Yeah. So these things can never be NP, assuming NP is not equal to P. These programs can never be general purpose because they can't allow me to solve these NP complete problems in polynomial time. So if they succeed in polynomial time doing anything, it's on a subset of those of the sequences. Yeah. So I think that makes sense that would guide us in considering these problems. All right, Isabel.
Yeah, I have a quick question. So I was wondering, when you were saying like naturally occurring versus random, like how random? Like how do you distinguish like randomly occurring from natural?
Yes, well, in this discussion, I've used the idea, basically there's naturally occurring, which we do understand. You found it in nature. And then by random, I mean, let me make it definite. Let's say a random amino acid sequence is you have a machine, I'm mumbling here, and it flips coins. Or it rolls dice. So the die that it rolls has numbers 1 through 20 on it. And it hits it's a fair die, hard to build in the real world, but it's a fair die, and it may come up on any of the 20 values. And when you roll it a second time, it's independent of the first time. Well, you just use that die to generate a sequence. And I claim, this is probably right, with high probability, the sequence you get, the primary sequence you get, will never fold in your lifetime. Okay? So that's the deal. Okay, so I think that's a theme that underlies all of these recent developments. Okay, and so just a brief story. This is an aside, but I can't get it out of my head. There was this, it's no easy thing getting even a protein that folds to form large enough crystals to X-ray crystallography them. Okay. And so it's weird stuff like, I've been trying to get this here protein, which folds very well by itself, but I want it to come together and form a crystal structure with other similarly folded things. A lot of it used to be that to get the thing to fold was magic. I mean, to get it to crystallize was magic. And this guy, Ned Seaman, told me that, sometimes it was that some pickle juice from a delicatessen down the street got into the solution, and then it crystallized. And no other pickle juice would work, right? It was that kind of science. And Ned, an X-ray crystallographer, was particularly unlucky in this regard because he never got anything of interest to crystallize. And he reasoned that crystallographer, no crystals, no crystallographer. Okay, that he was a professor and he wouldn't get tenure if he couldn't crystallize anything. So he went in another direction, and he's one of the primary figures in what's now called DNA computing and DNA self-assembly. Okay. Anybody interested in doing preen theory? Okay. Yeah. All right, good.
If a society's membership drops to zero, that society has gone extinct, but what happens to the cultural preens? They may survive. One way for a cultural preen to survive the extinction of a society is to be stored in physical things that remain after the society has gone. For example, it is currently thought that the society, perhaps that of the Essenes, that produced the Dead Sea Scrolls may have been extinct for about 2,000 years when the scrolls re-emerged in the middle of the 20th century, and some of the cultural preens they stored were resurrected as cultural preens of modern societies. For example, many recent translations of the Bible rely on the Dead Sea Scrolls as a primary source.
Right. So even though the Essenes, their society went away long ago, we nonetheless have a lot of their cultural preens have outlasted the demise of the society that created them. All right.
As an aside, it appears that Bach's Brandenburg Concerti preen was not stored in even one brain for over a century. However, the cultural preen was not extinct. It was stored in a manuscript in the archives of the Margrave of Brandenburg, where it remained, spore-like, until about 1849, about a century after Bach's death, when it was rediscovered and published. Cf. Beowulf.
So if you happen to know and like the Brandenburg concertos, they almost did not make it. By the way, they were almost victims of the printing press extinction because the Brandenburg concertos existed as a manuscript, but they weren't printed. Okay, here we go.
Another way that a cultural preen can survive the extinction of a society is to migrate to another society before the extinction. For example, when the Western Roman society ended in 476 CE, by convention, many of its Christian cultural preens had already found new homes in the cultural preensets of other Western European societies.
Okay, so, the Roman society officially ended, but of course, by then, its cultural preens included many Christian ones, and those survived because they had already made it to other societies. Okay.
Notice that genetic preens can also survive the death of a species. We saw that the genetic preens of smallpox viruses escaped to computers just before all copies stored in members, at least in the wild, of the smallpox species had perished.
Right, the species went away, but there were copies of the genetic preens now on computers, right? So those preens had escaped the species.
It seems possible that some of the genetic preens related to photosynthesis that arose about 3.5 billion years ago have survived through species after species and are now present in modern cyanobacteria.
Okay, so it's about 3.5 billion years ago that we first saw photosynthesis occur. Okay. And it's an incredibly amazing and complex process by which, photons are converted into chemical energy, ATP usually. And those things 3.5 billion years ago presumably are gone now. But we still have all sorts of plants that still use photosynthesis. Now, it could be that some actual gene sequences of considerable length have made it from 3.5 billion years ago through numerous different species to still be around today. Get to that image. Okay, so
This long-term persistence of genetic preens and cultural preens, while species and societies come and go, is part of what makes these preens more fundamental objects than species and societies.
okay, so for me, that's an important point. That is, societies come and go. So do species come and go. But these preens march right through them. Not all of them, they eventually go too. That is, they reach copy number 0. But they can persist for presumably, certainly thousands and probably even billions of years by bouncing from species to species or society to society or whatever. Okay? And that's why they're more fundamental. So I wasted a lot of time making images. And let me see for a second. Okay. Making images with Gemini and ChatGPT. So let's talk about biology. So each of these balls represents, bubbles represents a cell, let's say. And all the little colored balls inside these bubbles are physical objects inside. Now, some of those physical objects are storing preens. In fact, they all are, but some of them are storing genetic preens. Yes, pieces of DNA and stuff. Okay. Now, this is an image of when a bubble, a bacterium, say, a cell, replicates. It does it like this, yes? And it's on its way to this replication. Okay, so kind of a simple image of cells and the stuff they have inside. But remember, among the things they have inside are some things that are storing genetic preens. A lot of things that they have inside aren't storing genetic preens. For example, pieces of sugar, right? All right. Now, okay. Here's another image of my attempt to get AI to do the right thing. Anyway, okay. So what's being shown here? Anybody? If you can parse
Apoptosis?
this. No, not apoptosis, because that's when a cell kills itself. But this big bubble in the middle.
Would that be the nucleus?
Not my intent. Okay. But, see, AI only works,
I'm like
with
really
that.
curious what prompts you used.
To use AI?
Yeah, to
Well,
generate.
because my artistic skills are, somewhat below novice level. Okay. And so anybody got an idea of what I intended here? Strange question.
Is it meiosis or mitosis?
Good guess, Ryan. Not correct. But, here's what I had in mind. This is a cell that's eaten another cell, right? I mean, could be. You see, it's got its own material inside, but now it's got this other thing it's eaten with its material inside. Yes? Okay. So, um, so, um, why does one cell eat another cell?
Well, this is actually one of the theories, as I'm sure like many are aware of how mitochondria arose in eukaryotic cells, for example, that
Yes,
they were endocytosed billions of years ago,
exactly.
and then became integrated into the replicatory structure of the cell. And then obviously, as the cell divides with mitosis, then some mitochondria are found in the new cell and then replicate themselves.
You're absolutely right about the mitochondria. I mean, that is the prevailing theory, and I don't see any reason why it can't be correct. However, most times when one cell eats another cell, endocytoses it, it's that is not going to be the outcome, that it starts living in, harmony with the larger cell. What is the more common thing that happens to a cell that's been eaten?
It dies. Mm,
Well, yeah, it's going to die.
yeah.
But why did the big cell bother?
I'm not a biologist, but if I had to guess, resources.
Yes, yes, right? That other cell has sugar and proteins and things like that. Now, I want to look at the little cell and all its resources. So if you take one resource, let's say inside the little cell was a protein. What will happen to that protein once the big cell gets around to it? You guys need to brush up on your biology, except Martino, who seems to know what's going on. Yeah. So what's going to happen to that protein?
Like, does it dissolve or it gets
Well,
dissolved and then it gets recycled, right? Or
yes, one could say it gets metabolized, right? So the big cell will typically have a bunch of enzymes that are specifically designed to tear apart proteins and to capture their amino acids. I suppose that in some cases they actually capture energy too that might have been stored in the chemical bonds. Martino, do you know any of the details of this?
Well, yeah, I mean, as far as I understand, what you're explaining is what I've been taught, like the existing proteins are depolymerized and then used to synthesize new.
Yeah. And what happens to the sugar that was in the little cell?
I'm
Oh,
not aware.
okay. Well, if it has sugar, it'll be metabolized to get the energy and the chemical bonds of the sugar, probably into the form of ATPs. which are sort of the energy currency of the cell, they're sometimes called, right? That is, this, the physical things inside the little cell are going to be used, recycled if necessary, into the big cell, right? Okay, and that includes, there'll be DNA strands in the little cell that are storing genetic prenes, right? And that DNA will also be broken down. Okay. And so the genetic prene it used to store has lost a copy number, right? Okay, so that's what happens. Now, in my imagination, we're going to go through this again. And it's all going to work out great. Okay. So what we're going to do is, now the bubbles are societies. Okay. And societies are full of physical objects. And some of those physical objects are storing cultural prenes, right? And some of the physical objects, store energy, and they can store, I guess, value, like pieces of gold and stuff. Yeah. Okay. Now, I'm not sure that societies divide in this way. I can think of one example where we might say this has happened, but I don't think it's particularly common. Okay. Does anybody have an example of a society dividing like a cell does?
That would be like when a colony declares independence, like the United States.
Ah, Ryan Rubel. Yeah. Yes. Yeah, that's kind of interesting. So what happens? Yeah. Now, no, there's something called exocytosis, as I recall. Am I right, Martino?
Yes.
You're the go-to guy here. Okay. And that's where a cell will sort of bud off a bit of itself and float it away. Usually, I think of that as being waste material, but it sounds like that exocytosis might be like your revolutions, insurgency revolutions, depending on which side of the coin you're on. Okay, and so it might be. I was also thinking that about when the Roman society split itself into the Western and Eastern society. So when that split occurred, on the day it occurred, I think that the Eastern and Western Roman societies probably had largely the same princess, for example. But of course, once they divide, they start to evolve on their own and do crazy things, and eventually they go their own ways. Okay, so now this is society. So in this view, societies are full of physical things, and some of them are storing cultural prenes. Yes. Okay. Among the things that a society will store are or have inside it are human beings. Yes. Who themselves store lots of cultural prenes in the form of memes and all sorts of bacterial genes and all sorts of stuff, right? Okay. Okay, so now let's talk about this image. Now, what is it showing? Because the bubbles are now societies.
That would be conquest.
Conquest, yes, Ryan, very good. And I also like the word conquest. So, like the cell, when one society conquers another, what's it going to do to the resources of that other society?
Probably claim them.
I'm sorry, what?
Probably claim them, although I think, I guess depending on what their um, purpose is for the conquered society, they may leave them with a subset of those materials. Otherwise, they're just kind of condemning them to death.
Yeah, good point. So I think you're right. This could be, for example, if you want, you can think of this as what Russia's trying to do to Ukraine. Again, that's a political matter, and string theory doesn't care. Okay. But you could think of that, or you could, for example, think of colonization, right? Of, the British around 1800 colonizing everything. Yeah. Now, when this happens, the material inside the smaller ball is physical objects. Well, in some cases, be metabolized. So, for example, they may tear down buildings of the smaller society and use the materials to build buildings they like. Here's another example. What if the smaller society has a gold statue of its famous leader? Yes. Then it's storing a cultural preen, right? Statues of famous. Okay. What will happen to that statue? Maybe. Lots of things could happen. What might happen to it?
You'll get melted.
Bingo. And it'll get melted, right? Thus destroying a cultural preen. And further, if we take this a little further, what might be done with that gold? And now I'm just sort of playing a game, but what might happen to that melted gold?
Right, reform into a statue of the leader of the conquering society.
Yes, it might. That's where I was going, right? Okay. And one sees such things a lot if one looks in history. Yes. Okay. Now, here's a good question for you. Among the physical things in the smaller society were human beings. What's going to happen to them? What could happen to them?
Subjugation.
Yes, subjugation. But let's talk about that a little more. What forms of subjugation could those take?